Moving on, we get to the second function in the list, which is energy transfer. Living organisms require a lot of energy to support their life processes, and they obtain this energy from their environment. Virtually all organisms can obtain energy by ingesting foodstuff molecules and degrading them and oxidizing them to carbon dioxide while obtaining energy. Some organisms like plants and algae and even some certain bacteria can also derive energy directly from the sunlight. But in either case, the energy is obtained in the form of high energy electrons. That's the first step. And the energy of such electrons is then stored chemically in the form of ATP molecules. ATP is a nucleotide. It is constantly being produced inside cells as a energy store, and it is constantly used by energy requiring processes. So whether the electrons and the energy are obtained from foodstuff molecules or from sunlight, the molecular systems that are responsible for such processes are very complex, and they are heavily based on proteins. So if we start with the first process of getting energy from foodstuff, this process happens inside, most of it happens inside cellular organelles called mitochondria. And that's why we often refer to mitochondria as the powerhouses of cells, because they specialize in the production of ATP. And the process actually starts outside mitochondria. It starts in the cytosol, where the foodstuff molecules are partially degraded and oxidized. And then the degradation products, as well as the high energy electrons that were obtained in these processes, are transferred into the mitochondria, where they are, where the degradation products are fully degraded and oxidized to carbon dioxide while giving away all their electrons. So the high energy electrons are then transferred into a chain of proteins that reside in the inner membrane of the mitochondrion. And when the electrons go through the proteins, they give away their energy. The energy is then channeled in a very complex way to a protein called ATP synthase. And this protein knows how to use the channeled energy to construct ATP molecules. So that's the first system. And as I said, in the case of plants and algae, there is also a second system which knows how to obtain energy directly from sunlight. And this system resides not inside mitochondria, it resides in other organelles called chloroplasts. And that's one difference. Another difference is that the goals of this process are different. The process, which is called photosynthesis, uses the energy to construct, not to synthesize ATP so much, but rather to construct glucose molecules. And they are then used as building blocks for, or as sources of energy for the plant. So the second system is also very complex, and it contained, a part of it knows how to channel the energy of the sun into a water molecule. The energy is used to split the water molecule and extract its electrons, and the electrons are then transferred through a similar chain of proteins. And again, the energy that is obtained from such a process is used in this case to construct glucose molecules. And this process is called photosynthesis. So we have two different systems that transfer energy and use it, but they use it for different goals, but both of them are still heavily based on proteins. The third role of proteins that we'll discuss is the import and export of solutes across membranes. In order to function properly, proteins need to exchange material with their environments constantly. For example, they need to import or internalize foodstuff molecules into the cell, and export waste products. In the case of certain specialized cells, they also export enzymes and hormones. The problem is that many of these molecules contain partial or full charges, and this makes their partitioning into membranes or crossing membranes a very energetically unfavorable process. And later on in the second lesson, we'll understand the physical basis for this phenomenon. So cells had to develop means to facilitate this transfer of solutes across membranes, and they found two main solutions. The first, in both cases, the solutions involve proteins. The first solution involves proteins that contain hollow pores inside them, and these pores are filled with water, and they allow ions and small molecules to go through the pore. So this way, the molecule that crosses the membrane doesn't feel, so to speak, the lipid membrane around it. It just goes through the water pore of the protein and gets to the other side. Such proteins are called channels. We can see two examples of such channels. This one transfers potassium ions through its pore, and the other one transport water molecules. And you can see more clearly here that the pore inside the protein is filled with water molecules. So channels are very prevalent in the membranes of cells, but they allow the transfer of solutes only from compartments in which their concentration is high to compartments in which their concentration is low, because this is a spontaneous process for material to go from high concentration to low concentration. And this type of transport is called passive transport. Besides channels, we also have another type of proteins called transporters, which we don't see here. Transporters or carriers, and these do not contain pores inside them. What they do is that they bind the solute on one side of the membrane, change conformation, and release it at the other side. Now, many transporters also mediate passive transport, like channels, but some of them can use external energy, usually in the form of ATP, but not always, to pump the solute against its concentration gradient, which means they transfer the solute from low concentration to high concentration. And this is called active transport. So these are the two solutions adopted by cells to transport ions and small molecules with charges across membranes. And we'll discuss it further when we get to the seventh chapter of this course. The third role in our list is cellular communication. Cells are able to communicate with one another, and this happens between unicellular organisms, but also between cells that are part of the same multicellular body. Much of this communication relies on chemical messengers, which are organic molecules that are produced by the source cell, secreted to the external medium. They then diffuse to the target cell, bind to a cell surface receptor, and this binding conveys the message to the target cell and change its biological activity. One example of such communication is in the nervous system of animals. In this case, the two cells that communicate are very close to one another, and the chemical messengers are called neurotransmitters. Another example is also in the animal body. This is the endocrine system. And in this case, the two communicating cells are very distant from one another. And the chemical messengers are called hormones. So because the two cells are distant, the only way the messengers can affect the target cell is to enter the bloodstream and travel through the circulation and get to the target cell, which again can be very distant. So how does this signaling process work? As we saw, the first event in this process is the binding of the chemical messenger to a cell surface receptor. This receptor is a protein, and once it binds the chemical messenger, it induces a small but distinct structural change in the receptor, allowing it to bind to an element inside the cell. This element is also a protein, and the binding to the receptor activates it, which allows it in turn to activate or inhibit the following element. And this goes on and results in the activation or inhibition of many elements inside the cell, most of which are proteins. These proteins can be enzymes that accelerate metabolic reactions. They can be transporters on the surface that allows the cell to import certain ions or molecules or export them. And they can even be proteins that go to the nucleus of the cell and bind genes on the DNA and change their expression patterns. So the conveyance of this of the signal resemble a relay race in which one element activates or inhibits the other, and so on. But in fact, it is more like a cascade, because many of these proteins, once activated, can activate a lot of other proteins. And this is what makes the whole process be very extensive inside the cell and lead to the change of its biological activity. This can end in proliferation of the cell, growth in size, differentiation, production of certain metabolites, and even in very extreme cases, it may cause the cell to commit suicide, a process called apoptosis. Here we can see an example of a signaling process which is very common in living organisms. It is based on a protein inside the cell called the G protein. The name comes from the fact that this protein is able to bind the small nucleotide GTP. And the receptor involved in this system is called GPCR, which stands for G protein coupled receptor. So the process starts with the chemical messenger binding to this GPCR. The chemical messenger can be a hormone or a neurotransmitter. As I said, this process is very common. And once it binds to the receptor, a small change in the receptor allows it to bind the G protein and activate it. And this results in a separation of one chain of the G protein, the one that binds the GTP, from the other two chains. And the activated chain then binds to an enzyme called adenylyl cyclase. This enzyme is able to convert the ATP molecule into a cyclic form called cyclic AMP. Cyclic AMP is a secondary messenger, which is a name that we give to small organic molecules that are involved in signal transduction inside cells. And the word secondary is meant to distinguish them from the primary messengers, which are the neurotransmitters or hormones. So once cyclic AMP is formed by the adenylyl cyclase enzyme, it binds to another enzyme called pKa. pKa stands for protein kinase A. And it is a protein able to carry out a process called phosphorylation, which is the attachment of a phosphate group to certain amino acids in target proteins. Now, pKa has a low specificity, which allows it to bind many target proteins. And by attaching a phosphate group to them, by phosphorylating them, to either activate them or inhibit them. And this results in many changes in the cell. So this is a very well-known and common example of a signaling process inside cells. Finally, many cells are able to communicate by direct attachment to one another instead of releasing chemical messengers that diffuse between them. In such cases, the attachment between the two cells is mediated by two receptors at the surfaces of the cells, one on the signaling cell, the other one on the receiving cell. And the two receptors bind to one another when the cells are close enough, and they convey the message. Now, the binding between the two between the surface receptors is usually very specific, which means that the receptors adapted throughout evolution to recognize each other in a very specific way. This type of direct binding between cells via their cell surface proteins also plays a role in defense, which is the next protein function that we'll discuss. And we have an example here of how this direct binding allows the immune system of animals to recognize foreign bacteria that invade their body. So when a bacterium invades an animal body, it is recognized by a certain type of defense cell that swallows this bacterium and degrades it into smaller components. Some of these components are peptides, which are specific to the bacterium. And these peptides are then presented onto the surface of the defense cell when they are bound to a surface protein called MHC2. When the foreign peptide is presented in that way, it can be recognized by an immune system cell called T lymphocyte. It is possible because the T lymphocyte has a receptor called the T cell receptor, which knows how to recognize specifically the complex of the foreign peptide with the MHC protein of the first type of cell. So this is a direct binding that mediates a recognition between two defense cells. And once this happens, the T lymphocyte becomes activated, and it alerts other branches of the immune system, allowing the system to mount an immune response against the specific bacterium from which the peptide was derived. Now we can see that in addition to this complex, the T lymphocyte also has another protein called CD4, which is able, again, to directly bind to the MHC2 protein. And this further facilitates the activation of the T lymphocyte. The irony is that while certain membrane proteins are important for defending the organism, because as we saw, they help the organism mount an immune response against an attacking pathogen, there are also proteins in the membrane of the host cell or the host organism that are exploited by viruses to get entry into the cell and infect it, and actually infect the whole organism. And this is because viruses have co-evolved with their target organisms, which means, among other things, that these viruses have developed certain proteins that are recognized by the host's membrane proteins, or some of them. And this recognition allows the viruses to bind to these membrane proteins, and this way become very close to the host cell and infect it. And we can see such an example here in the following image. We chose the coronavirus. You probably heard about it. It's responsible for the latest epidemic, the COVID-19 epidemic. And if we look at its structure, we see that it has a lipid envelope. And through this envelope, there are different proteins that protrude and face the external environment. One of these proteins, the one shown in green, is called the spike protein or S protein. And this spike protein has evolved to be recognized, but not one, but two membrane proteins of the host cell. The first one is called ACE2. This stands for angiotensin converting enzyme type 2. And this enzyme is a regular protein in the host. It's responsible for, or it's actually part of the system that regulates blood pressure in animals. But anyway, this receptor, this protein, recognizes the spike protein, allowing the virus to get close to the cell. And then the other protein, which is also an enzyme called TMPRSS2, actually cuts parts of the spike protein, and this activates a process that allows the virus to fuse, actually allows the lipid envelope of the virus to fuse with the membrane of the cell, and this way expose the inner environment of the virus to the inner environment of the cell. And as you probably know, if you studied immunology or virology, this allows the virus to hijack the different machineries, molecular machineries inside the whole cell, and by this way to replicate and to reproduce. Now, this image is a little misleading because it implies that the fusion happens on the surface of the cell, but actually what happens is that once the virus binds to ACE2, the cell uses the membrane to envelope the virus and internalize it into the cell inside a vesicle called an endosome. And only then the fusion occurs and the virus can go on with its life cycle. So this is an example how certain membrane proteins are actually a liability to the cell in the sense that they are used by viruses to attack the cell and to infect the whole organism. Finally, we have proteins that form large structures, both inside and outside the cell. And we can see these structures in the image here. The intracellular structure is called the cytoskeleton, and it is made by proteins like actin, myosin, and tubulin. These proteins form large fibers. They assemble into large fibers that go throughout the cell. The other structure, which is outside the cell, is called the extracellular matrix. And it is formed by proteins like collagen and elastin. We'll discuss it in more detail in chapter 2. Now, you can see that the two structures are connected to one another by proteins, some of which cross the cellular membrane. And this allows changes, for example, in the extracellular matrix, to be conveyed into the cell and be felt by the cytoskeleton, and sometimes even affect processes that occur inside the cell.